Welcome to Microbiology. This is going to be Chapter 1 screencast, and the screencasts are going to be in parts. So usually I'll say Part 1, Part 2, and so on, depending on how far I get, because I try to keep them short, 15 minutes or less. You can see a picture of the book here, Microbiology Fundamentals, A Clinical Approach by Cowan and Smith. It's the 4th edition. I do recommend that you use that book. And of course, as I've mentioned in the orientation information, you do need the access code in order to complete all of the activities in Connect, which is part of this, this whole book. So I want to point out that while I do my best to make sure that I only post PowerPoints that are identical to my screencast PowerPoints, there are times that as I'm going through the PowerPoint before I record my screencast that I might throw out a slide or maybe add a little blurb to a slide. And I apologize for that, but I'm constantly trying to improve and make things better for this class. So if you notice that something isn't exactly right in the PowerPoint that you printed in comparison to the screencast PowerPoint, it's best to rely on the screencast PowerPoint and, of course, the worksheets that I've posted. So always revert to the screencast PowerPoint that I have recorded. All right, so on to Chapter 1. So microbes, they are tiny. They're teeny tiny little things. Can't see them without some sort of help, okay, like a microscope. Microbiology is a specialized area of biology, deals with living things, teeny tiny. Different microorganisms include bacteria, archaea, protozoa, fungi, helminths, algae, viruses, and prions. The main topic we will talk about in this class is bacteria, and we will also have an entire chapter devoted to viruses, which are incredibly interesting. And it's probably, I guess, scary to some people at the same time. But the more we know, the better we'll be informed. And then we'll talk a little bit about, just barely a little bit on archaea. And then we'll talk a little bit about protozoa, fungi, helminths. And we'll mention prions down the line here in this class. Chapter 1 is kind of an overview of the whole book and everything that we're going to cover. So Chapter 1 can feel a little kind of disorganized and confusing, but just know that we're gonna dig deep into these topics as we move along in this course. So microbes can be easy and also difficult to study. They're easy because, generally speaking, they reproduce rapidly. We can grow them in the lab, but we need special tools to be able to study them. So that's why they say they're easy and difficult. When I say that they reproduce rapidly, I will say this, that if you've ever had a culture done, a wound culture or a urine culture, it's going to be at least 24 hours before you get those results, depending on what type of culture it is. Wound cultures can take, 24, 48 hours before a definitive result comes back. And then there's the time for the results to get to the doctor from the lab and so on. And then those results have to get from the doctor to the patient. So all in all, it can take several days to get these results. Generally speaking, I think that probably in the next 10 years, we're going to be moving towards more genetic testing. They're already starting to do genetic testing to identify microbes. It is more definitive. It's quicker than waiting for that 24 to 48 hours. It's still kind of expensive right now, but it's going to get to be normal practice, and the prices will go down for that. So microbes and the planet. So microbes have been part of what has shaped the Earth's habitat. Okay, we're talking for billions of years. 3.8 billions of years ago, the single-celled organisms supposedly appeared on this planet, and it is amazing how they can figure these things out, but they have. And one of the things that they've discovered is that cells arose from a single, now extinct, common ancestor, and the cells such as eukaryotes And prokaryotes arose from that single-celled common ancestor. Eukaryotes are cells with nuclei or a nucleus. Like our cells, I guess you could call us our cells are eukaryotic. Prokaryotes are single-celled critters, single-celled organisms with no true nucleus, as in the definition of what we know a nucleus to be. There are statements that it is like a nucleus-like object in some of these cells, but they're not referring to it as a true nucleus. So I know that your slide is a little different from this one, the one that I posted, so beware of that. And again, you will focus on what I have here in this recording versus what's on that slide that is posted in the class. So this is just a slide showing you the relatedness of organisms. We have that last common ancestor, and then that's how all of these three things kind of branched off. I'm not going to get into this. And actually, let me go back to this slide just for a second. So eukaryotes have a true nucleus, and then bacteria and archaea both fall into the prokaryotic category. So they are both referred to as prokaryotes. All right, so bacteria and archaea versus eukaryotes. Eukaryotes, many are single-celled, but they aren't all. They have the ability to develop into highly complex multicellular organisms, hence the human person. The eukaryotic cells are larger in size than prokaryotes, and eukaryotic cells are a small minority compared to bacteria and archaea when you're talking about counting up all of the cells. There are way more bacteria and archaea than there are eukaryotic cells. When you think about how many cells we have, that's just kind of incredible. So ubiquitous is a term to basically describe everywhere. So microbes are everywhere. They are found in the Earth's crust, polar ice caps and oceans, inside the bodies of plants and animals, in the Earth's landscape, and including places that we haven't even discovered. And also, it's important to note that microbes are essential to life. As you know, if you disrupt your normal biota, your normal flora in your body, it can cause a complete shift and cause some issues. So, for example, if you take an antibiotic and you can end up with a stomach issue where it's causing you to have loose stools. And when that happens, it's because your normal biota in your gut is disrupted by that antibiotic, which is basically wiping out all the good stuff, all the good bacteria that's in your gut, because it's wiping out the stuff that needs to be there. And that will disrupt all of that and cause havoc. As we know, many of us have experienced the fun times of taking antibiotic and having other secondary issues from that. Theories of evolution in science. So theory of evolution is the accumulation of changes that occur in an organism as they adapt to their environments. They're documented every day in all corners of the planet. And these theories of evolution are actually testable by science. Theories in science have undergone years and years of testing and have not been disproved in a well-studied and well-established natural phenomenon, and they are not just a random guess. So science is science, and that's that. Historical uses of microbes by humans. Obviously, if you make bread, you use a little bit of yeast to make some bread. You go to Texas Roadhouse and have, those yeast rolls that they bring to your table. Those are really good. That's part of the deal. These microbes help to make bread. Obviously, alcohol, cheese. Sometimes there are microbes used to treat wounds and lesions. There are microbes used to mine precious metals. I haven't really learned too much about that, but it'd be interesting to research. And then, of course, cleaning up human-created contamination. So we have, oil-eating bacteria. We have all sorts of things that are used to help get rid of what us humans have done to the earth. So biotechnology, when we're talking about microbiology, I mentioned earlier that they do some genetic testing to help to diagnose certain microbes causing infections. With that comes lots of other kind of areas in the micro world, one of which is genetic engineering. So we can manipulate the genetics of certain microbes in plants and animals for the purpose of creating new products and genetically modified organisms. And, I'm not going to get into a big debate about GMOs, and, everybody has their thoughts on GMOs, but what I understand about GMOs, what I've learned about GMOs over the years, is that they're not necessarily changing, they're not making, killer cantaloupes, right? They're making a cantaloupe that, if they genetically engineer a cantaloupe, they're making it so it's going to, withstand mold, right? So, or better yet, strawberries. It's strawberry season while I'm recording this, okay? And how many times have you bought a thing of strawberries from the grocery store or from a U-Pick kind of farm, and you get it home and you sit it on the counter and you forget about it for 2 days, and you go back, you dump them into a bowl to rinse them, and they're covered in mold. And for me, I would love for those to not be moldy that quick. And so scientists can manipulate the genes in plants to withstand mold, which is, kind of a cool thing, because now food can last a little bit longer. We're not spending more money on food and such. So that's my take on that. Obviously, creating a plant that, creates a Jack and the Beanstalk idea is, a whole different story. But recombinant DNA technology makes it possible to transfer genetic material from one organism to another and deliberately alter the DNA. Bioremediation uses microbes already present or introduced intentionally to restore stability or clean up toxic pollutants. I think this next one should be here also. Nope. Okay, so I will stop this recording here, and we'll pick up on part 2, chapter 1 in the next recording.